All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Today we have a really exciting drug to discuss, and that is Renitradine. Now, remember, Renitradine has been used for Helicobacter pylori, or the heartburn or the gastric ulcers, and has been banned in the US and Europe because of the discovery of some of the carcinogenic material in it. However, there is a study that looked at the Ranitidine Bismuth Citrate and looked at how it can actually inactivate the replication or stop the replication of coronavirus. So that is a discussion that we'll do today. In my opinion, it is a very similar discovery like we saw with ivermectin. When ivermectin in the beginning was proposed, people were actually dismissing that ivermectin is not going to be able to be useful. And as we have been seeing that it is actually very useful. Similarly, the mechanism here that I would discuss with you, I think you would agree that this has a potential as well. Now, this drug has not been used or trialed in humans. It has been tested on SARS-CoV-2 infected cells, and it has been tested on mice that were infected with SARS-CoV-2, and it was able to reduce in vitro, that means in the lab, the infectivity of the SARS-CoV-2 by 1,000-fold and by 100-fold in the SARS-CoV-2 infected mice. So I thought this is a really interesting one to discuss. And before we discuss that, I wanted to give you an update on on 3 patients that I had been talking about. So the first one, my own family member who we thought had COVID because of the symptoms were very similar to COVID. They got their RT-PCR and they were negative. So all of us who were quarantining and who were kind of staying safe and staying away, we are all back to normal. So that is one update. Thank you very much. In the meantime, that I had many people send their prayers and good wishes. So thanks for that. Second is the Cool Bean I discussed, whose age is 58. He is, I believe, from in an African country. He's from India, and he had developed COVID as well, and his RT-PCR was positive. So his latest is this. So today, I had put him on ivermectin. He was already taking hydroxychloroquine, plus he was taking some prophylaxis as well. So today's update was, and it's about 6 or 7 days. I had 99.5 fever in the morning, but after the paracetamol, it has been normal all day. I have worked all day from home and even went for a 30 minutes walk in the evening. So I would say that I am much better. Oxygenation during the day was 97%. So this is the outcome. And I feel that he had mild symptoms as well and has recovered or will recover in another day or so. Then a third Cool Bean. He has been a big friend of mine for years now. He is a doctor in Greece and has been watching my immunology lectures and is very, very much interested in immunology and mechanisms as well. So today, he sent me this note. By the way, I had COVID already 6 days. The 3 first days as a carrier, and then from Friday on had full-blown COVID up to 39.5 degrees Celsius for 6 hours, 3 hours interval and temperature. So he had the temperature. He did not have other symptoms. Used mouth gargles as you proposed, iodine ones twice a day. When it felt like itchy after a couple of hours, it went away. Oxygen saturation 98%, no respiratory sounds, no dyspnea, no nothing. And he's a doctor who is actually leading in a hospital. He is leading the COVID unit. So of course his exposure is greater as well. And then he says that some brain fog for 1 day. I used Math Plus protocol, but with Mobeen's Extra Plus. So I used hydroxychloroquine and zinc as well. 48 hours, that's all it took. And now I am in quarantine, and I also continue prevention from possibility of another phase or something until my B lymphocytes create the proper antibodies. So he's waiting for the adaptive arm to become active, although his innate arm or the early phase has helped. Of course, I did all the prophylaxis discussed online by you beforehand, and so I was ready to beat it in the beginning. My vitamin D levels should be around 80 nanogram, if not more, and zinc I take every other day all the time. So this is the, so Jim, you were asking, do you know what the D3 is? So he suspects that his D3 is about 80 nanogram. So I have been promising you about the prophylaxis for some time. I think we should be able to put that together. My challenge is that every day when I start the morning, I start preparing for the lectures, and I don't find enough time to put together a document for the prophylaxis. So maybe if you are comfortable, I can maybe take a day off from teaching and then just use that day to put together the document, and then we'll go back to our normal process. So with this, I'm pretty happy for all these three cool beans that they are all doing well and happy. And now let's start our discussion. So this is drbeen.com. Here is the article, or actually a study. This is October 15th, so pretty recent, University of Hong Kong, and I would be discussing this one. There is a link in the description to this study. Then there is another study here. I believe it is from the same team. This is also SARS coronavirus 2, NSP13. There is a protein NSP13 possesses NTPases and RNA helicase activities that can be inhibited by bismuth salts. So that is what I'm going to be explaining today. The takeaway right in the beginning is that bismuth salts can be useful to inhibit the replication of the SARS-CoV-2. So that is the basic outcome of this whole talk today. Then there is another study here from 2012, an older study that was about the SARS-CoV and the helicase in SARS-CoV. So that was the first outbreak. And then they have explained the mechanism of function of the helicase. So if you're interested, please look into that as well. And finally, there is a diagram here which I want to use for the for explaining where the NSP13 is. Again, credit goes back to the original authors and drawers of these diagrams. This is not my diagram. So with this, and I guess you know my diagrams are mostly cartoonish. So here, Ranitidine Bismuth Citrate. The takeaway or the discussion today that you have to keep an eye on is the helicase. What is a helicase? What does it do? And how are its functions disrupted by bismuth? That is a discussion. So first of all, the bismuth is going to act as a virostatic. That means it would reduce further increase in number of viruses. It can be called antiviral in a broader term, but antiviral will mean something that would kill an existing virus. This would not kill a virus. However, when a virus is replicating, when it is increasing in numbers in our cells, it would reduce the replication count or the numbers. So if you look at this over here, this is a helicase. I made it like a crane with a drill on it. Helicase normally in the viruses can do a lot of functions, but two major functions that we want to discuss today, which are relevant to SARS-CoV-2 section, are number one, they help in RNA unwinding. And I'll explain what does that mean. There is the genome of the virus, when it is replicating, that genome would need unwinding, and we'll talk about that. And the second is when the virus is replicating in our cells, for the replication process, energy is needed. So the virus helicase, this protein, this enzyme from the virus, would break down our energy that our cells made for ourselves and break it down and use it for its own replication. So in this regard, I also want to comment on this, that sometimes people reach out to me and say viruses are dead, they do not use energy, they do not move and all that. That is correct. Viruses are dead. And it's always a problem with the medical sciences to say, how do we address them? How do we talk about them? At the same time, when a virus enters our cell, the proteins of the virus are going to hijack our cell and then start using our machinery. And for that usage, for that machinery to operate, energy is needed. And the virus is going to use our energy to do its own functions. So that energy usage is called NTP hydrolase. Nucleoside triphosphate hydrolase action is where we break, for example, adenosine triphosphate or ATP or GTP and others. In case of SARS-CoV-2 helicase, it primarily breaks ATP, which is our energy source, and then uses that for its own functions. So now let's very quickly see what is the function of helicase. So I'm going to do a high-level discussion of the replication of the virus, how a virus, when it has arrived in our cell, would increase in numbers. So imagine that this is inside the cell we are looking in. So this is the original virus genome or RNA that has entered into our cell. We call it a positive sense single-stranded RNA. So what that means is, for the medical folks here, 5' end is here and 3' end is here, and the ribosome would attach on this side. This is what we mean by a positive sense. And the positive sense RNA is already ready for our ribosome to act on it and to start transcribing or translating. So here, if you see in the next step, once the virus is in our body, in our cell, then the RNA of the virus is available. That RNA will hijack or will get connected to our ribosome, which is our ribosome uses our RNA to produce proteins for us. But now the viral RNA is here. It's going to get connected to our ribosome and make it start making viral proteins. So this process is called translation, and I have done a detailed discussion about this. That is why I'm not going to go too much in depth here. But now here is the ribosome, which is our machinery. It is being used by the viral. It is using viruses' RNA to produce virus proteins. A couple of proteins that are really important are RDRP. RDRP is RNA-dependent RNA polymerase, or simply, as I've made it look like a photocopying machine. Imagine that this enzyme of the virus is going to create photocopies or the copies of the virus genome or virus's RNA. So it is absolutely necessary for the virus to make more viruses to copy its brain, which is its RNA. It also makes helicase. So helicase, once again, is that enzyme of the virus, which is going to break our energy sources, ATP, to use for its own function. Plus, at the same time, helicase's function is to open up or unwind the RNAs as well. So we'll look into that. So these two enzymes are now available. Then what happens is, if you see here, this is the helicase. What helicase is going to do is, in the first step, it is going to start breaking our energy sources. And so if you see here, nucleoside triphosphate, these are 3 phosphates. And I've discussed this in the past, that phosphates are like currency inside the cell. These are the energy sources. If you have 3 phosphates, you are a rich man inside a cell. If you have 2 phosphates, you're slightly poor. And if you have 1 phosphate, you are really poor. So here what happens is, nucleoside, for example, let's say ATP. ATP, TP means triphosphate, adenosine triphosphate, so it has 3 phosphate attached to it. The virus helicase comes around and it breaks one of the phosphates away. When it breaks it, that liberates energy. That energy is going to be used for the virus functions. So this is one function of the virus helicase. Then, now this is the RDRP, which is the second enzyme of the virus. What that is going to do is, it's going to pick up the original virus RNA, and it's going to make a copy of it. So here is the copy strand. Once these strands are formed, now can you see that there are actually 2 strands that are formed and joined together? We need to now peel them away from each other like Velcro. We need to separate them. And to separate them, once again, helicase would come in function. And what it would do is, it would use its enzyme area to kind of separate the 2 threads that are connected together, the RNA, and unwind them and help them separate. So imagine now if the helicase was not functioning, then this viral RNA replication will not occur successfully, because these two, the original RNA and the new copied RNA, are stuck to each other, and they need to be separated to become more copies and to become part of the new viruses. But if they cannot be separated, they're not going to make new viruses. Similarly, if you go up here, if the helicase is not going to break energy bonds and make energy available, then the viral creation process would stop. All right, so now we have helicase here. It has separated out the newly formed RNA and the original RNA. Then, and check this out, in the helicase, if I make it slightly bigger, inside the helicase, there is a area, this is an enzyme, correct? In that enzyme, it's a protein. In the enzyme, there is an area where a metal is attached, and that is zinc. It uses zinc to perform its functions. And the Ranitidine Bismuth Citrate, what that does is it has bismuth in it, which is also a metal. It will replace this zinc with its own bismuth, which would cause the helicase to become ineffective. So we replace zinc from the virus and we add a bismuth in there. So we add a wrong part in the machinery of the virus. So we throw a wrench in its engine. And now the virus machinery, helicase, cannot function anymore, because where it needed zinc, over there instead there is a bismuth. That is the eventual goal for today's discussion. All right, so now we have the RNA copy produced. That RNA copy is separated by the helicase. The new RNA that is produced is called a negative sense RNA. What does that mean? It is a copy of the original RNA, but just like if you stand in front of a mirror, the copy in your copy in the mirror is actually flipped around. And to make you again, what would happen is we'll need to put another mirror in front of that mirror to make it the orientation corrected. So same way, once we have made the copy, this copy has the wrong orientation of the nucleic acids on it, nucleic acids. So we call it a negative sense RNA. For the medicos and health professionals, what that means is that 3' end is on this side and 5' end is on this side. And this 3', 5' end is not useful to attach to a ribosome and to make proteins. So what does the virus do? The RDRP then makes another copy of this new one. And when this new copy, the black one over here, when it is made, then the 5' end is back here and 3' end is back here. And now we have a positive sense RNA again. So this is a process that continues and repeats to make more and more RNAs, which will become packaged in more and more viruses. So once another copy is made, then that copy again, these two guys need to be separated as well. They're also stuck with each other like a Velcro. And guess who would come in? Once again, Helicase, which is a viral enzyme that would come in and separate them as well, like a zipper to unzip them. Once these are unzipped, this new positive sense RNA that is formed will take part in making viral proteins, plus some of it will be preserved to go become part of the new viruses. Now, some part of this new RNA that are formed are these are called subgenomic RNAs, or they're smaller pieces of RNA, and they would help make more proteins. Virus, the SARS-CoV-2, has many proteins. For example, it has the spike protein. Then it has the N protein, which is nucleocapsid protein. Then it has M protein. It has RDRP. It has many proteins in it. So these smaller portions of RNA are going to then help make more proteins of the virus. Eventually, all of them will come together and will be assembled in a new virus. So for this whole process, the virus will continue to need more energy. So Helicase not only is separating the newly formed or unwinding, I should say, the newly formed RNAs, but Helicase is continuing to make more and more energy available as well. And so then those little genomic sub-RNAs, will continue to make viral proteins. Eventually, those viral proteins will come together and these will be assembled in the endoplasmic reticulum and then Golgi apparatus, and a new virus or a bunch of new viruses will be formed. Now, the cat is always calling me. So now we have the function of ranitidine, and this is the last point for today. What ranitidine bismuth citrate does is it replaces zinc with bismuth. So if you see here, there is bismuth in there. Once bismuth is in there, this enzyme stops functioning. And once it stops functioning, this is as you saw, it is a critical enzyme for viral genomic replication. This enzyme can actually help with many other viral protein functions too, but that is not the focus of my discussion today. So generally here, once the helicase has become ineffective, it cannot make more energy available, that is one. And secondly, it cannot separate out the threads, the newly formed RNAs, and the virus replication will stop. And so now let's look at the studies for a second. First of all, what is the NSP13? So this helicase is formed by the genome of the virus, the SARS-CoV-2, by a specific area of the genome called NSP13. So let's very quickly look into that. So here, if you see, this is the virus genome, and there is an open reading frame 1AB. Open reading frames are the frames in the genetic material of the virus which do not make things that are part of the virus assembly. Instead, they make tools that would help assemble the virus. So the virus enters our body, makes its tools to make more viruses, then packages the viruses together. So open reading frames are usually the one that make tools instead of the proteins that will be packaged in the virus. So here, if you see, NSP13 is over here. I know it is very small, but NSP13 is over here. This is part of the subgenome, which is called PP1AB. So just some technical information for medicos in there and researchers. So now let's look at very quickly a summary of what they have found. It is really interesting. So this team from Hong Kong University said that a commonly used anti-ulcer drug is what they used. They had seen that this drug in the past had been useful against viruses. Plus, remember, this is used for Helicobacter pylorus, which is a bacteria which causes the ulcers, and it is used against that as well. So they suspected that this might have something to do with the SARS-CoV-2. It might be effective there as well, just like ivermectin folks did it. So what they did was they took this NSP13 protein, which is a helicase, and they put the ranitidine bismuth citrate on it, and that caused, look at this. So RBC is ranitidine bismuth citrate. RBC has been demonstrated to greatly reduce viral load by over 1,000 folds in SARS-CoV-2 infected cells in the lab. In particular, in golden Syrian hamster model, RBC suppresses SARS-CoV-2 replication to reduce viral load by 100-fold in both the upper and the lower respiratory tracts. So that is a very, very good thing. RBC exhibits a low cytotoxicity. So the cytotoxic effect, meaning if we take it, would it start causing our cells damage in the dose that is necessary to reduce the viral replication? So they say again, this is not a human trial, but from their study on the infected cell, they saw that the dose that is given is safe. Then here is the mechanism of action. It irreversible means that once it has removed the zinc from the helicase and replaced that with bismuth, this helicase has become useless now. It can never become corrected again. There are some enzymes that if you modify them, they would very soon become corrected once again. So this is destroyed. It would not be corrected. Now this cell would need another virus to come in and bring more helicase for these helicases to function. So it irreversibly kicks out the crucial zinc ions in the zinc binding domain to change it to bismuth bound via a distinct metal displacement route. So he's talking about helicase and talking about the action of the RBC on it. And this is the compound. NSP3 helicase and potent inhibited both the ATPase, so the energy generation function, and the DNA unwinding function. They both are destroyed at these doses, 0.69 micrometer and 0.70 micrometer. So that is great. And that is the discussion for today. I actually think that this is a pretty exciting drug as well. So with this, welcome back from the weekend. Please like, subscribe, and share. If you wanted to donate and support the work that I'm doing, there is a link in the description, and you can use that for donations. And I'm just going to quickly look at the thread here, and then hopefully we break and we'll meet again tomorrow. So Arun says, bismuth will replace all zinc necessary for our metabolic activities? No. In the Ranitidine Bismuth Citrate would selectively remove zinc from the helicase, and ranitidine has been known to be safe to us for decades now. The only thing is now that there are some carcinogenic particles found in it. Because of that, it is taken off the shelf. But the zinc part, it does not affect our cells because we have been using it without that disruption. So good question. Southwest says, if I don't see your show today, I got to be pissed. Well, you have Luffy here as well. We are both here to keep you happy. So Guy Teffler says that so every replication doubles. Ideally, what happens is normally, so I just made one thread. Normally, the original genome comes in, the virus's RNA, then it is copied into a negative sense RNA. Then that negative sense, and many copies are made, thousands and millions of copies. Then those negative sense RNAs are further copied again or transcribed again, and these would then start becoming part of the new viruses. Plus, some of them will be used up by our ribosome to make virus proteins. So it's not necessary that it is a doubling action. It's just a continuous action with our ribosomes that we will just keep duplicating them again and again and again. Ocean says that if you did telehealth, I would. I don't. So one, I don't practice in the US. Secondly, I do practice internationally. Thirdly, I only practice in more charitable way. I never charge fee. So that is my way of practicing. My primary function is that I run drbeen.com. And so this practice is just, this is my contribution. Arun says that, is coronavirus a single-stranded RNA? Correct. It is a positive sense single-stranded RNA. However, when it arrives in our body and our polymerases, RDRP, and then our polymerases, primarily RDRPs, when they work on it to make copies of it, that is when it becomes a duplicate strand, not duplicate or double helix. And for that helix to be separated, for the newly formed copy to be separated, we need helicases. Cool. So I think these are the main discussion points for today. eDog says that brilliant stuff, Dr. Bean. See you tomorrow. France, absolutely. So see you guys tomorrow. And thank you very much. Once again, please like, subscribe, and share. And I would see you tomorrow.